Hello again. I had to chop that mutation one into two. So this one is about how prokaryotic cells can have mutations happen like we talked about, but they can also take parts of their DNA, either plasmids or their chromosome, and they can transfer it to an already living other one in 4 different ways, and therefore give this a new combination of genes than it had before, and therefore some different traits. So this, hope you like that music in the background. So this is about recombination, all right? And remember, these guys, they don't do meiosis, so they don't have crossing over and random lining up of chromosomes. They don't do fertilization. Those are the three kinds of recombination events that happen in eukaryotic organisms like ourselves. These are prokaryotic, except for, as we'll see, two of them also apply to eukaryotic cells. So here we go. So now they throw in some genetic recombination in prokaryotes. Remember, in sexually reproducing eukaryotes, we had crossing over, independent assortment, the random lining up of chromosomes and meiosis, and random fertilization. But since none of those three events, right, or two events, meiosis and fertilization, happen with prokaryotes, right, we see some different things that have evolved, and some of those also apply to eukaryotes like you and I. So here we go with that, right? Here's the blue box where they name them, and they call this horizontal acquisition of genetic information. And what that means is, instead of passing these on to their offspring, which they'll eventually do, these changes are going to get passed on to an already living bacteria cell, therefore horizontal. Now, when this changed bacteria cell has kids, we kind of generally refer to it as descent, right? They're descendants, kind of down, descend. That's why they chose this term horizontal for this. Okay, there's 4 different ways that bacteria cells can have their DNA, and therefore any kind of trait resulting from them, get into another prokaryote and therefore cause genetic variation, increasing variation. Okay, one of them is that DNA might just leak out of this and into that. The uptake of naked foreign from a different one DNA, that's transformation. Transduction is when a virus infects this cell and picks up some of its DNA, and a new virus with that new DNA deposits it in this cell. This is getting a little messy, right? That's called transduction. That is one of the ones that can also happen with eukaryotic cells. In fact, quite a bit of your genes or your DNA in general have come from past viral infections in your ancestors. So they threw this little box in here. Related viruses infecting the same cell, right, might combine or recombine genetic information and then pass that recombined stuff on. I don't know why they need to put that in there, but you get the idea. The other two ways, right, are conjugation. This is conjugation where you have this actual tube form between two prokaryotes. And this one, the one with these pili, none of that made the blue box, right? Pili, whatever. This one is going to send a plasmid, that kind of DNA, to this other cell, right? It's actually usually going to send a copy of the plasmid. You can see that here. So now this cell has the plasmid, whatever genes are in there, and this cell now has some new traits, okay? This one is purely prokaryotic, right? And then you have transposition, the last one, which is also eukaryotic and prokaryotic, and that's where genes might get up and jump. That's why these are also called jumping genes, from one place to another, maybe from a plasmid to the bacterial chromosome, maybe to the chromosome to a plasmid. That recombined plasmid might later get to another bacteria cell by conjugation. So these four things can kind of interact as time goes by to create lots of variety. So we have two more topics in this last unit, and they're biggies, so it might be more than two PowerPoint note slides. But so yeah, I was at like the other side of this bar. Neither one of those is me, and the bartender out of nowhere says, "We don't serve time travelers here." Then a time traveler walked in. That's a good one. Well, that's not so good. That's bad. Anyway, have a good day.